Hello my dear students, welcome back. So in the last video I started about the endomembrane system, and in the endomembrane system I told you total 4 cell organelles are there which are its constituents, that are endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. And endoplasmic reticulum we finished in the last video. Now in this video we are going to see about the Golgi apparatus, or it is also called as Golgi bodies. This word has came from the name of a scientist, Camillo Golgi. Okay, so Camillo Golgi was the first scientist to observe this cell organelle. That way the name is given Golgi apparatus or Golgi bodies. So this is the structure of the Golgi apparatus or Golgi body. Clear? So in this structure, two phases are there. That is very important. You can see this one phase and this is the second phase. This phase is called a cis phase or convex phase or forming phase, and the other one is called as trans phase, concave phase or maturing phase. Before I discuss between these two phases, you should know that what is the function of the Golgi apparatus or Golgi body. So the chief, or we can say the main function of the Golgi apparatus or the Golgi body is in the packaging. Means whatever the packaging is done inside a cell, that is done by the Golgi apparatus. What happened? Suppose the formation of protein or lipid take place inside the endoplasmic reticulum, but before it is transported outside the cell, or whether it is used in the at any intracellular receptor, that substance is to be packaged. For example, just I want to tell you, suppose if anything is manufactured in any company, before the marketing, or we can say before it is delivered, it is to be packaged. Similarly, if there is any formation of any metabolic substances inside a cell, then it is to be packaged, and the work of the packaging is done by this Golgi apparatus. Clear? Means this cell organelle will perform the function of the packaging. So you can say this is the cis phase, convex phase or forming phase, and then trans phase, concave phase or maturing phase. These two are two distinct phases of the Golgi apparatus, and this cis phase and trans phase, they are connected to each other with the help of parallelly stacked structures. They are called as what? Cisternae, or it is also called as Golgi cisternae. Okay, so the cisternae are the parallel stacked structures which connects the cis phase and the trans phase. So what happens? Any substance that may be either protein, lipid or steroid which is to be packaged, that will enter through this way, through this cis phase or the convex phase, or we can say forming phase. Means it will enter through this way, then it will pass through different cisternae, and as it will pass through different cisternae, it will become mature, and then it will be delivered out through the trans phase, concave phase, or we can say maturing phase, in the form of vesicle. This here vesicle I have shown. So what is this vesicle? This vesicle is lipid, protein or steroid or any other metabolic substances which is enveloped by the membrane, because that packaging is done by the Golgi apparatus. Clear? Now what is the location of this Golgi apparatus? So inside the cell, this Golgi apparatus is found mainly at two places. It can be either present near to the nucleus, or maximum chances are there that it may be present very close to the endoplasmic reticulum. Why it is present in the close proximity to the endoplasmic reticulum? Because whatever the substances that is formed by the endoplasmic reticulum, the packaging of that particular substance is done inside the Golgi apparatus. So that this is the reason why the Golgi apparatus is in the close proximity to the endoplasmic reticulum. Clear? So this is all about the Golgi apparatus. So you may write down Camillo Golgi first observed Golgi apparatus near the nucleus. It consists of many flat disc-shaped structures called this one, that is called what? Cisternae. That is called cisternae. And you can see the number of cisternae can be variable. So varied number of cisternae are present in the Golgi complex. Varied number of cisternae are present in a Golgi complex. There is not any particular number, so varied number of cisternae can be present. Golgi cisternae are concentrically arranged near the nucleus with distinct cysts or forming or convex face, and you can write concave, trans or maturing phase. So this Golgi cisternae have these two phases. So even though these two, you can see, they may appear different, but they are attached to each other with the help of this cisternae. Clear? Now you write down what is the function of cisternae. I told you, or sorry, what is the function of Golgi apparatus? So the main function of Golgi apparatus is in the packaging material. So the Golgi apparatus perform the function of packaging materials. That material can be any metabolic substance. Okay. Now I told you the material which is to be packaged, that will enter through the cyst phase or the convex phase, and then it will pass through the cisternae, and then it will be released out through the trans phase or the maturing phase in the form of vesicle. So the materials which is to be packaged enters through the cyst phase, and after passing through cisternae comes out through maturing phase, maturing or trans phase in the form of vesicle. Clear? Now one more thing you should know. I told you that this Golgi apparatus are found in the close proximity, or we can say in the close association with the endoplasmic reticulum, because the proteins and lipids which are synthesized by the endoplasmic reticulum, they are modified in the Golgi cisternae before they are released from the trans phase. So this is the reason why the Golgi bodies are in the close proximity to the endoplasmic reticulum. So Golgi apparatus are present in close proximity to the endoplasmic reticulum because the materials which are synthesized by ER are modified by Golgi cisternae before it is released from transphase. Okay, so this is the reason why the Golgi apparatus are in the close contact, or instead they lie in the close proximity to the endoplasmic reticulum. So this is all about the Golgi apparatus. Okay, now the next cell organelle is very simple, lysosome. So this lysosome is also a membrane bound cell organelle, and this lysosome is also called by a special name that is also called a suicidal bag, or it is also called as digestive bags. Why they are called so, that I will discuss. Actually, this lysosome is a single membrane bound cell organelle which contains all the hydrolytic enzymes like lipases, proteases and carbohydrases. Okay, means it can digest a variety of the substances. Okay, so this is the special feature of the lysosome, that it contains all the hydrolytic enzymes. Then why it is called as suicidal bags? What happens when the cell becomes very old? Now it is the cell decides to suicide itself. So the suicidal of the cell takes place with the help of lysosome. What happens? The lysosome will burst out by breaking out its membrane. So all the components of the cytoplasm will come directly in the contact with the lysosome. Therefore, it will dissolve all the components of the cytoplasm, and as a result the cell will die. That's why it is called as suicidal bags. Then why it is called as digestive bags? Obviously, as a variety of digestive enzymes are present in them, therefore they are called as what? Digestive bags. Okay. So lysosomes, they are single membrane bound cell organelle. They are single membrane bound cell organelle which are formed by the process, this is very important, which are formed by the process of packaging, in Golgi apparatus. So it is very important that this formation of lysosomes take place from the Golgi apparatus as a result of the packaging. Lysosome contains variety of hydrolytic enzymes, like lipases, proteases and carbohydrates. That's why they are called digestive bags. That's why they're called as what? Digestive bags. And then again, I told you they are also called as suicidal bags because in the worn out cells, or we can say in the old cells, this lysosomal contents are able to dissolve all the components of the cell, or we can say they are able to dissolve all the components of the cytoplasm. That's why they're called as suicidal bags. In worn out cells, the content of lysosome help in dissolving the constituent of cell, in bracket में written suicide. Therefore, it is also called suicidal bag. It is also called a suicidal bag. This much you should know about the lysosome. And then the last component of the endomembrane system is vacuole. So I told you earlier that all the components of the endomembrane system, they are membrane bound, that we have seen. So this vacuole also, it is a membrane bound cell organelle. It is a single membrane bound cell organelle, and the membrane is given a very special name. That name is tonoplast. So the tonoplast is exclusively the membrane of the vacuole. Clear? Then about this vacuole you should know. Actually, this vacuole contains all the materials which are not useful for the cell. Okay, not that much useful, like water, sap, and excretory waste product. Okay, so that is the function of the vacuole. Means it will act as a storage cell organelle, and mainly it stores the water, sap, and excretory waste. One very important thing you should know, that this vacuole is very bigger inside in the case of plant cell. It may occupy around 90% volume of the plant cell, but it is very much less present, or we can say it may be absent even in the case of the animal cell. So vacuole is single membrane bound cell organelle, single membrane bound cell organelle which contains water, sap, and excretory waste and other materials not useful for the cell. Clear? Then I told you the membrane of this vacuole is very special, that is called as tonoplast. The membrane of vacuole is called, this name is very important, tonoplast. So it is called as what? Tonoplast. Then one more important aspect of vacuole I told you. In the case of plant cell, this vacuole is very big and it may occupy around 90% volume of the cell. Vacuole is well developed in plant cell, occupying around 90% volume of cell, while in animal cell it is very small or may be absent, or may be absent. One more aspect of this vacuole you should know, that they are actively, means they are very much important in the case of this osmosis, means we can say endosmosis and exosmosis, and that we will discuss in the that plant physiology. So here only basic aspect of vacuole I am discussing. How the transport take place through this tonoplast, that we will discuss in the plant physiology. So this completes the endomembrane system. Okay. Now from the next video I will start the mitochondria. Thank you.